Before sexual reproduction, a parent must produce gametes in the body before the haploid cell meets with the other parent's haploid cell. This process is called meiosis, which is split into two processes, meiosis I and meiosis II.
The whole process of getting ready for cell division, which in this case is meiosis, is interphase. Interphase duplicates the 46 chromosomes to make 46 chromatids, which are joined by the centromere. A big chunk of the cell cycle is interphase. Meiosis I is one of two processes that split a parent diploid cell into 4 haploid cells. In prophase I, the nucleus disappears and spindle fibers form. The centrioles, which were outside in the first place getting ready for cell division, move in and make the spindle fibers. In metaphase I, the sister chromatids line up side by side with their pair in the center of the cell. It goes through crossing over before the spindle fibers attach. Anaphase I is the phase where the chromatids are pulled apart to the poles. The spindle fibers pull the chromatids apart toward the centrioles. Telophase I is preparing for the intercytokinesis, which is the same as cytokinesis, but is the one of 2 times the cell is going to split, and is where all the chromatids get surrounded at the opposite ends with the nucleus that forms. Intercytokinesis is where for the 2 separate cells get separated in the middle, but is the transition for meiosis II. It is the halfway point of meiosis. Each cell now has 23 chromatids.
Crossing over and independent assortment occur to make variation in each gamete. This process occurs in meiosis I. Crossing over is the exchange of genetic material between homologous chromosomes with different coding, which means that sister chromatids don't exchange genetic material. This happens during metaphase I, when the chromatids are lined up in the center. This is why all babies, even siblings, don't look completely alike. Independent assortment arranges pairs of chromatids randomly, resulting in an infinite amount of possibilities. Lining up homologous pairs and switching the chromatids inside will result in genetic diversity and the variation of gametes. Independent assortment occurs once each time one phase transfers to another, from prophase I to metaphase I to anaphase I, so a total of 3 rearrangements. Meiosis II is basically 2 cells going through mitosis simultaneously, resulting with 4 haploid gametes. In prophase II, in both cells, spindles form and the nucleus disappear. Centrioles from the duplicated cells create the spindle fibers for the chromatids. Metaphase II, in both cells, have the chromatids line up in the center, and the spindle fibers attach. In anaphase II, the chromatids are pulled apart in each cell and form the gametes with the chromosomes. In telophase II, the nucleus in both cells form and start to pinch together, forming a shape reminiscent of 4 cells. Cytokinesis, the final process of meiosis, is where the 2 cells split into 4 haploid gametes.